So glycolysis is regulated at three primary places: the regulation of the enzyme hexokinase or glucokinase, the regulation of phosphofructokinase 1, and the regulation of pyruvate kinase. These are three irreversible steps in glycolysis. Pyruvate dehydrogenase complex is also regulated, but we'll address this regulation in the next module that covers the TCA cycle. So the regulation of hexokinase or glucokinase is twofold. First off, the product glucose-6-phosphate can inhibit the activity of the enzyme, such that as glucose-6-phosphate elevates in the cell, it will inhibit the kinase and glucose will be left in the bloodstream. Glucokinase is also regulated by a regulatory protein. And in the absence of glucose or fructose 1-phosphate, glucokinase is bound in the nucleus to its regulatory protein. The regulatory protein also enhances the transcription of glucokinase. In the presence of glucose or fructose 1-phosphate, glucokinase is released from the nucleus and can be active in the cytosol. This translocation is facilitated by both glucose and fructose 1-phosphate. The second regulatory step in glycolysis is by the enzyme phosphofructokinase 1. Phosphofructokinase 1 is activated by high levels of AMP and high levels of fructose 2,6-bisphosphate. This enzyme can also be inhibited by ATP and citrate. So, the activation of this enzyme by AMP is very logical. When a cell does not have very much energy, that stimulates the cell to oxidize glucose to generate ATP. The high levels of fructose 2,6-bisphosphate are a little different. Fructose 2,6-bisphosphate is generated by the phosphorylation of fructose 6-phosphate to the 2,6-bisphosphate by an enzyme phosphofructokinase 2. This compound is not considered part of the glycolytic pathway. Rather, it's a shunt of fructose 6-phosphate. So in the case of an elevation of fructose 6-phosphate, this is an alternative pathway for that product. Fructose 2,6-bisphosphate in turn stimulates the activity of phosphofructokinase 1, increasing the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. In this case, fructose 6-bisphosphate acts as a stimulator to the enzyme. The enzyme phosphofructokinase itself is a bifunctional enzyme with two domains. It has a kinase domain, as we've just seen, that can phosphorylate fructose 6-phosphate to the 2,6-phosphate. When the kinase domain is active, the enzyme is referred to as phosphofructokinase 2, or PFK2. The enzyme also has a phosphatase domain that removes a phosphate from fructose 2,6-bisphosphate. When the phosphatase domain is active, the enzyme is termed fructose bisphosphatase 2. We'll discuss the activity of this enzyme in a few modules when we look at the pathway of gluconeogenesis. The inhibition of this pathway by ATP and citrate are also fairly logical. If the cell has enough ATP, then it doesn't need to oxidize more substrate. Likewise, citrate, we'll see, is an intermediate of the TCA cycle that can be shuttled out of the mitochondrion and used for the synthesis of fatty acids. So if citrate is accumulating within the cytosol, it will also reduce the oxidation of glucose. The next regulatory step is that of pyruvate kinase. Pyruvate kinase is stimulated by fructose 6,1,6 bisphosphate and again inhibited by ATP. So as products of the pathway of glycolysis begin to elevate, they'll stimulate the activity of lower portions of the pathway. The regulation of glycolysis and all of the other pathways we look at is critical to understanding the rest of intermediary metabolism. Again, these steps within glycolysis are not reversible. So there are other functions of glycolysis besides generating and reducing equivalents for electron transport. First off, glucose 6-phosphate, as we've said, can go to glycogen synthesis, or it can generate 5-carbon sugars via the pentose phosphate pathway. And we know that 5-carbon sugars are required for the synthesis of nucleotides. It can generate 2,3-biglycerol phosphate, which is a regulator of hemoglobin. This influences the binding of oxygen to hemoglobin. Several intermediates can also result in amino acid production, the generation of both serine and alanine. The acetyl-CoA generated by glucose catabolism can be used to synthesize fatty acids. Remember, this portion of the cycle is within the mitochondrion, and what comes out here is citrate, and citrate is used to synthesize fatty acids. Similarly, glycerol, a 3-carbon molecule, is used as a backbone in the synthesis of triglycerides, which combines 3 fatty acid chains and a glyceride molecule. The TCA cycle can also generate glutamate and other amino acid intermediates. So let's look at this question. When fructose 2,6-phosphate levels are elevated in the cell, which of the following is a true statement? Glucose levels are high. Yes, I would say that's true. Remember, fructose 2,6-phosphate is generated by the phosphorylation of fructose 6-phosphate to fructose 2,6-phosphate when the 1,6 molecule is elevated. PFK2 will be activated. I would again say this is true, right? Fructose 2,6-phosphate stimulates the activity of PFK1. Pyruvate kinase will be activated. I would agree that this is also true. Pyruvate kinase is stimulated by high levels of fructose 1,6-phosphate, and in the case of a situation where fructose 2,6-phosphate is elevated, the 1,6-phosphate is probably also elevated. D. NADPH production is increased. This is also true. Elevation of fructose 2,6-phosphate suggests an increased flux through glycolysis and subsequently the increase in the production of NADH. So in this case, all of these are true statements. Let's look at that again. Where here, fructose 6-phosphate can be converted to fructose 2,6-phosphate by the enzyme phosphofructokinase. This stimulates the activity here, and the pathway moves in the forward direction. This generates elevated levels of NADH, which can be oxidized in the electron transport chain.